Good afternoon and welcome to the latest Yellowstone Advisory webinar with Christian Hoyer Millar, Director and co-founder of Oxford Biodynamics. Christian is going to give an introduction to the company and will update on the most recent publications. So many thanks to everyone for joining the webinar today. You're all currently on listen-only mode, but if you want to ask a question, please type into the chat box at the bottom of your screen. We've had a few questions sent in ahead of time and we'll cover all questions at the end of the presentation. The format today is a presentation from Christian, which will last approximately 30 minutes, and then we'll have 10 to 15 minutes available for Q&A at the end. And so just before we start the presentation, I'm going to put up on screen a poll. And if I could ask you all to complete that poll, and once that's completed, we'll come on to the presentation. Well, thank you very much for that. Looks like we've got a split audience of roughly 50% holders and 50% non-holders, so that's interesting. I'm now right. I'm just going to stop sharing now, and just to let you know that following the meeting, you'll be redirected to a short anonymous survey, and it'd be really appreciated if you could spend a few moments completing this. I think that's the housekeeping done for. So without further delay, I'm really pleased to hand you over to Christian Hoyer Millar, Director and co-founder of Oxford Biodynamics. Over to you, Christian.
Thank you, Alex. Good afternoon. Thank you very much for joining the webinar. I'll give you an introduction over the next 20-30 minutes on OBD, and as Alex said, some recent updates. For those of you who don't know the background to OBD, it's an Oxford University spin-out, and we listed on AIM in December 2016. We've raised about $40 million to date. And our message to the market is that we are the leaders in what's called 3D genomics, worldwide leaders. And we're really the leaders for two reasons. First of all, we have a very strong patent position, which is shown in the appendix to this presentation. And secondly, we have significant know-how in how to look at these 3D genomic loci. We're located in 3 different places. We have effectively a blood collection resource in Penang in Malaysia. We have a significant laboratory in Oxford, and we have a growing position in the States. We've worked on over 30 plus indications, which I will show you some of them over this presentation, in oncology, autoimmune, neurodegenerative and metabolic. And we used to say that we didn't work in infectious diseases, but as you'll see later, we're putting our first toe into that marketplace with our latest announcement. Importantly, we're on 3 NIH biomarker steering committees. These are the committees in the States mandated by the government to inform pharma effectively of the importance of biomarkers. And we are on 3 committees: oncology, immunology, and neurodegeneration. And as you'll see, we've worked with most of the large pharma or biotech companies, especially in the States. We've got a strong balance sheet. As of the 30th of September, we've got 15.5 million in cash. Our burn rate is relatively low at 3 to 4 million a year. on a net basis. So what we develop are biomarkers, and biomarkers are really any reading of a particular condition. So a biomarker for dementia could be a memory test. In our case, our biomarkers are all from whole blood. This is something that was not acknowledged as possible a few years ago, but is now known as liquid biopsy. So effectively, we can see any condition systemically in blood on a very low volume of blood. And those are the questions that we're addressing. I mean, most people don't really understand that the failure rate on drug development is greater than 90%, and that 90% of all drugs only work on 30 to 50% of the people. So if you can develop biomarkers that indicate who might respond or not respond, that's a huge asset for any pharma or biotech company. So biotech and pharma are using our biomarker signatures to work out which patients respond and how do they respond, and also to improve their clinical trials program in terms of the outcome and comply with FDA requirements. If you know who's more likely to respond, you can, with the FDA's guidance, you can put those responders into your clinical trial and obviously make your chances of success in that clinical trial considerably greater. And also we're used by biotech and pharma to increase competitive advantage. Pharma wants to be able to say to the payers, the insurance companies or governments, that their drug works on a certain cohort of patients and therefore their drug should be bought. There are different types of biomarker, and we've worked in all of these areas. So there's obviously the predictive type of biomarker, which is to do with treatment. Does this drug work or not? Then importantly, there is residual disease monitoring. The patient has taken the drug, and does the disease come back again? Then there's prognostic biomarkers. Is the condition that the patient is under an aggressive or a non-aggressive condition? If you're running a clinical trial and you want to see if efficacy, you want a cohort of patients in your clinical trial who've got the most aggressive form of the disease, so that you can see the response or the non-response as quickly as possible. And then there are response biomarkers. We all respond differently to different drugs. And you need to know, as a clinician, whether the patient is going to take 2 weeks to respond or a week in order to make the right clinical diagnosis going forward. The leadership that we have is in chromosome conformation signatures, which are known as CCSs. We have a proprietary position here in terms of our patent position, which is actually the first patent is owned by Oxford. And then we have a series of 14 different patent families, which have improved our intellectual property position considerably. Genetics is the study of the human genome, but to understand how the genome is expressed, you really need to understand how the genome is structured, which is epigenetics. So you can think about epigenetics really being the software and genetics the hardware. Or another way of looking at it is, if you play poker, the hand you get dealt of cards are your genes, but how you play those cards and how you interrelate with everyone else in that game of poker is epigenetics. And the value of epigenetics is increasingly acknowledged. And just a little bit on what we have, DNA is arranged in a 3D structure around chromatin called a CCS. And it's these CCSs that cause many of the interactions that take place within a cell, and therefore the condition of the patient. What we uniquely have is the know-how of where to look for these CCS changes. and to see where the most important CCSs exist. It's not just us, OBD, who says that this is important. This is quite a complicated picture. It was written out of by a man called Tordini in Cambridge. And basically what he's saying is that everything we read, all the modalities, the other ways you can look at patients, require an interrogation into chromatin conformation if you're going to stratify patients. So if you're going to tell red patients from green patients, you need to interrogate that blue box, the chromatin conformation box, and that's firmly where our IP is. Actually, it was understood quite a long time ago. If you look at the bottom right-hand publication, this was a publication from McGill quite many years ago, which basically said that chromatin conformation signatures were the ideal human disease biomarker. The publication on the left is the Harvard publication where OBD has written the whole chapter on epigenetics. As I said earlier, we're in a broad range of applications. In order to establish a position in a particular disease area, you have to have received the samples and you have to have worked on those samples. And when we started out, we had only used our technology in a very limited number of disease areas. We've now worked in over 32 different disease areas, and we are seeing the stratification as fundamental and very robust. As an example, we can't obviously tell you every single customer that we have. It's only when they put a publication in the public domain. You can see that in oncology, Pfizer, EMD, Mayo, Genentech, Prostagram in the UK. And there's a list of the various areas that we've worked on recently and the various customers. The process is relatively simple. There's a picture of our laboratory. There's no what's called the black box here. We know where to look for these markers. That's critical. And then we have a process which the industry understands using both PCR and qPCR that enables us to answer these stratifying questions. And of course, we've all been reading the papers in terms of diagnostics over the last 2 or 3 months. There are 2 areas that you have to have. One, you have to be sensitive. You have to have a test which is sensitive. Can you detect, for instance, breast cancer? And then you also have to have the same test has to be specific, i.e., are you telling someone that they've got breast cancer when they haven't? And it seems to us that over the last 2 or 3 months, there is suddenly a real understanding in the marketplace about the importance of having both of those as close to 100% as you can. Give you an idea on what we do and why we're confident about what we do. We've recently, as I suggested, signed up with Glasgow University really to look at 2 different things. One, to see whether we can stratify patients for response for this Japanese drug, which is licensed in Japan for influenza. It's actually a generic drug. It's on trial around in a number of different places around the world, in Italy, Mass Gen, and Brigham in the States. But importantly, to see whether we can isolate those patients who will suffer what are called cytokine storms. This is when your body overreacts and your immune system effectively, in the end of the day, kills you very quickly because of this hyperinflammation that these cytokine storms create. This is a serious problem with COVID. And because of the work that we have done, of which I'll show you some coming up, we're very confident that we will be able to tell doctors from blood, whole blood, on a stratified basis, those patients who might be affected by cytokine storms. In terms of our contractual position, this is this contract we announced last year with Mitsubishi in the States. This is for a drug, in fact, a drug that's now the FDA have asked to be taken to what's called phase 4. And the FDA have asked for biomarkers to be created by OBD to show effectively who will respond to a particular drug, who will respond to this drug or not. The two graphs at the bottom are why the FDA incorporated OBD into the study. The top one is the traditional way of looking at ALS patients, which is called an ALS-FRS score. So effectively what happens is that anyone with this terrible condition of ALS will go into their doctor over a 3-month period, and the doctor will use other biomarkers. How is the patient's breathing? How is he walking? Those sort of biomarkers to determine whether the patient has ALS. And it typically takes 3 months to determine whether the patient has ALS or not. That 3 months is a large proportion, unfortunately, of an ALS patient's lifespan. On average, ALS patients probably have only 2 years from diagnosis. So it's incredibly important to understand at an earlier point whether the patient has ALS. The bottom right-hand graph is exactly the same information from patients, but we were able to give Mitsubishi that information on a more accurate basis from blood on day 1. So the patient comes in, blood is taken, and we can tell what their diagnosis and prognosis is from day 1. This came out into the public domain last month, so it's new. This is work that Roche and Genentech published and is based on DLBCL, which is diffuse large B-cell lymphoma. And Genentech and Roche have 6 drugs developing in this area. But the problem is that there are a third of the patients, they can't tell whether they are poor prognosis, ABC, or good prognosis, GCB. So they effectively don't know what drug to give them. On the right-hand side, you can see that Roche and Genentech, in the bottom right-hand corner, tried to differentiate the patients in order to ascertain who these what condition these third of the patients were in, and they were unable to stratify the pink line from the blue line. Effectively, they're the same. What they published last month is that when they came to OBD and used our technology, which is called EpiSwitch, there was almost perfect stratification and differentiation between those who had poor prognosis, the blue line, and the pink line, which is good prognosis. Not only that, they showed this information against the competitors or indirect competitors of OBD, one called NanoString, which is a big company in the States, and another called Fluidigm. And the difference from the graphs is really that if you can see, there is an unclassified black line in both the NanoString and the Fluidigm graphs. Effectively, they couldn't tell what condition these patients were in, and there was no unclassified in the OBD materials, in the OBD work. So this was a very important publication for Roche to publish this very recently. the work that we published in December has led to a number of different important positions that the company has, contractual positions that the company is now involved in. And again, if we go back and think about the COVID situation, this is quite a good example of the power of the technology that we have. So if you look at the first graph, this is this is a graph showing the responders are against the non-responders for a particular anti-PD-1, PD-L1 therapy. This is an immunotherapy drug. And you can see that the non-responders fall off a cliff pretty quickly, and the responders are gradually dying out over a long period of time. But this is from clinical outcome. This is what actually happened to these patients. If you look at the right, on the right-hand side, we have almost a perfect replication of that from blood on day one. So in other words, the drug company, EMD and Pfizer, are able to tell who's going to respond or not respond to their particular drug. Seeing immunotherapy drugs are between $100,000 and $500,000 a year per patient. This is incredibly important information for the payers and obviously for the patients, because what you don't want to do is to give patients a drug that doesn't work, obviously. But in immunotherapy, there is also a condition which is different but similar to the cytokine storms that I talked about before, in that you get what are called super progressors. These are patients who, for a biological reason, die very quickly when you give them the drug. It's very similar to COVID. And this is a study to show how powerful really the EpiSwitch is. This is an independent study published at the end of last year in JAMA Oncology. On the right-hand side, you'll see all the indirect competitors to OBD. Some of them are huge companies. But what the graph is showing is in every published trial in the field of immunotherapy, how specific the false positive rate, or how sensitive the true positive rate were their tests. And if you add Oxford Biodynamics to that graph, you'll see that there are two things. One, we are the closest to the top left-hand side, which is 100% sensitive and no false positives. But also the number of patients and samples we've worked on, the size of the football, is far greater than in the other studies. So this has been very important because obviously this is independent validation, effectively, of the power of the technology. From a commercial point of view, there are really two or three things that are important. First of all, the FDA are increasingly asking drug companies to have biomarkers. Drugs are very expensive. Patients need to have the right treatment, and if there is the availability of an understanding or a likelihood of response, non-response, then the FDA is, in some cases, as in the ALS study, mandating that biomarkers are used. Our agreements are effectively on the basis that when our biomarkers are used, then those biomarkers are licensed, and there is a payment upfront, as upfront license fee, and then we get paid as the drug goes through clinical trial, and we receive a small royalty on the sale of the drug. In the case of ALS, of Mitsubishi, for instance, their sales are relatively small at 200 million. They believe that if they can get a wider label from the FDA, their potential market size a year is closer to $1 billion. So the benefit of using these biomarkers for a company like Mitsubishi is really considerable if they can get a wider label, an ability to prescribe that drug to a wider audience. And in the public domain, these are some representative deals which show some sort of scope for what these licenses might look like. So in summary, we are definitely the world leaders in 3D genomics technology. This is very important in terms of stratification. We have to get that messaging out. So that's why we are concentrating on building infrastructure, effectively people in the States. Most companies haven't heard of us in the States. And the ones that we're working with have kept most of the information that we've been working on confidential. This is an extremely robust and efficient technology, and we would expect to prove that yet again in terms of this COVID disaster that's going on now, in terms of a very important problem, these cytokine storms. And as I've said, there is a growing appreciation worldwide that the incorporation of biomarkers in drug development is critical, and therefore the value of EpiSwitch. As I said earlier, OBD has a strong balance sheet, unique IP and know-how, and as there is an increasing understanding of the importance of proper diagnostics, prognostics, which everyone is becoming aware of in the marketplace. So thank you very much. There are a couple of slides at the end in the appendices for you, which are to do with the IP, but I'm very happy to answer any questions.
Thank you, Christian, for that introduction to Oxford Biodynamics. Epigenetics and chromosome conformation signatures are a complex area, and you've explained them really well. I guess you can see how this technology is of huge benefit to patients, medical practitioners, and pharmaceutical companies. We've received a number of questions ahead of your presentation, and a number have come in during the presentation. And just as a reminder, if you'd like to ask a question, please type it into the Q&A, and Christian will try and get round to answer them in the time we've got available now. So just starting with some of those questions, can you give us some more information about the recent COVID-19 project? Are you getting paid? How long will it last? And how do you monetize this?
Well, it's a very good question. So yes, we are getting our costs. It was we've had a number of requests from a number of parts of the British government, even for our equipment over the last 2 months, for our robots. So it's been very difficult. The decision-making process for us has been quite difficult. So we are being paid our costs. The samples are being collected as we speak. The original plan had been that this would be very relevant for what the government considered to be the second wave in the fall, in the autumn. But of course, now we don't really know when the second wave it will hit. We should get preliminary readouts on this by the beginning of July, assuming the samples. We're not actually set up for infectious diseases, our laboratory in Oxford, which is why we don't do them. It's a different regulatory environment. So we've transferred the technology to Glasgow. So the work will be conducted there, and then we will be using our know-how in order to make this stratification. In terms of the second part of the question, yes, we think this is very, very valuable validation, not just for the utility in terms of COVID, but in a number of different areas. Sepsis, when we talked about IO, this ability for the body to hyper-inflame is a condition that happens in other areas. So we think that this is a very important validation step.
Great, thank you for that update there. Another question on one of the contracts that you signed last year. It's on the US Master Service Agreement. And the question is, have you started work on the US Master Service Agreement? Can you tell us which major US pharmaceutical you're working with on this? Can you provide any update on progress and what sort of contract has been signed? Does it involve an upfront payment, milestone payments, and royalty rates?
Gosh. So the samples have arrived. We were fortunate before the lockdown, and we are working on those samples. In fact, we're working. The laboratory is working on a sort of shift basis. I'm not. It's relatively easy to work out who that contract was with. But I'm not allowed to say who that contract was with. And no, I'm not allowed to give the financial terms, but it is a global deal for that large pharma.
Great, thank you very much. We've had a couple of questions on, I guess, director changes that have happened more recently. It's around, you announced a new sort of chief exec earlier this year. Can you talk about that appointment and how you'll work with him? And I guess, could you talk a little bit about the timing of that change, why it occurred now?
Yes, John Burrows came on board at the end of March. He is based in Maryland in the States. He is a prognostic guy. This was part of the strategy to effectively ramp up our influence and marketing in the States. Obviously, he is on lockdown, so he hasn't been here. Almost the day he took over was the day we locked down. So in terms of the division of roles, I'm still doing most of the things that he can't do because he's locked down. And the emphasis from him is commercialization in the States and setting up infrastructure in the States. And we will be dovetailing sort of executive parts of the business as and when he is able to visit the UK.
Okay, brilliant. Thank you for that. Another question here on one of the contracts that you mentioned. You mentioned that you're working with Mitsubishi Tanabe on one of their contracts. And the question really is, have they collected all the blood samples yet? Are you analyzing any blood samples? And how long does the contract have to run?
So the answer to that is these patients are very sick. So no, they're only halfway through their collection. And until there's a release of the lockdown in the States, which is obviously happening in some states, that blood has not been sent to us. When the blood collection is completed, we take blood on day 1 and we take it after 6 months. So the length of the study is 6 months.
Okay, great. Another question, you touched on this in the presentation about the excellent work you'd done and had been reported on in translational medicine. The question basically said it appears that this is an excellent study. It seems to suggest that OBD's technology is better at identifying DLBCL subtypes, better than Roche. Can you tell us, what does this really mean? And should this or could this have been announced as an RNS?
Yes, it's a very good question. So we thought about announcing it as an RNS, but we thought it would get swamped with all the COVID RNSs. So we may do a separate RNS. It is in the public domain, and we're thinking about how to do it in the States, because obviously Roche are a very important player in this marketplace.
Okay, thank you. I think you might have answered this, but I'll mention it again. It says, when can we expect data on COVID-19? I think you said July. And what are the attached commercial arrangements, which maybe you can't answer, but I'll leave that with you, Christian.
Well, yes. I mean, it's more perspective in terms of what will be the commercial arrangements. Our relationship with Glasgow has been going on for a number of years. They're a major, Professor McInnes is and Carl Goodyear are major supporters of our technology. And we have transferred the technology there. So we believe that this will be a very important development for OBD.
Okay. Question here. Hopefully you all know what this means, because I'm not sure I do, Christian. What is your opinion on silencing genes?
I'm not the right person to answer that question, so I'm going to pass on that one. Okay. And it's not fair. We often get asked questions on other other companies or their technologies, and I think it's not really right for us to comment.
Okay, fine. Looking at the US, what are the incremental costs, capital or operating, to the US expansion, and is there any visibility on a US licensing agreement?
Yeah, so John is working on what is required in terms of infrastructure in the States, and I'm sure he will report on that. I think it's people, not physical buildings at the moment, and he will definitely be reporting in when he can on progress in terms of contractual commitments.
Okay. And a linked question, which I think you've answered: what is missing from your US division? It sounds like you're, trying to bulk up the number of staff you've got out there.
Yes, it's getting our name around and getting people to understand that we exist.
Okay. Martin. A couple of sort of operating or more detailed operating questions. Can you discuss more about the robotics elements of proteomics, genomics, and are you also going down the specific route of sequenomics, and what is the forecast for this market over the next year?
So our we use Tekken robots. The advantage of Tekken robots is really twofold. First of all, that of course robots replicate the work exactly every time that they undertake the work. And secondly, our capacity has considerably been enhanced over the last 2 or 3 years. But I would think our capacity in terms of robots is probably quadrupled over the last 2 years because of the advance in robotics. So that's been a significant benefit to us. I'm not quite sure in terms of the turnover, I'm not quite sure what the second part of the question was really asking.
Okay. We'll skip to the next question, which is on cash and cash burn. You sort of mentioned a rate earlier in the presentation of 3 to 4 million a year, I think. Question is this a steady rate now, or could it step up? And if there is commercial, could it step up if there is commercial progress, or are these projects already funded? And at what point does the business become self-sufficient?
Yeah, so the business, I mean, clearly we will be building some overhead in the States, and the projections are for the business to become self-sufficient in the not too distant future.
Okay, thank you. I think you mentioned you had the sort of a slide on IP, and the question is, how much of your know-how is IP, and could a competitor develop a similar product using chromosome confirmation signatures that is sufficiently different to EpiSwitch to avoid patent laws, but would still compete against you?
Yeah. So I mean, IP clearly is very visible. And so there have been very large companies, some of whom have been on these slides actually, who have tried to file patents in our space, but because of OBD's prior art, they have been rejected. Absolutely, definitely, there will be, because we believe this is fundamental technology, there will be large companies who try and infringe on a patent side, on a know-how side, this is complex, and that is much more difficult. The know-how we have is very significant, but in terms of infringement, in theory, anyone who produces a product through interrogation of these CCSs potentially is an infringer. But of course, what you do is you wait until they infringe and they make money before you take any action. Okay,
yeah, that sounds makes sense. A question now on the share price. The performance over the last 4 years has seen it drop almost 75% before most recent recovery. What is new in the story now?
Well, I think what's new in the story now is that we have far more evidence than we did at IPO that this technology is fundamentally effective. There is a far greater understanding in the marketplace of the importance of biomarkers, and there is now a belief that you can get these biomarkers from what are called liquid biopsy. And I think now in 2020, there is a greater understanding of the importance of having proper diagnostics and proper biomarkers if you're going to deal with any of the awful conditions that exist. So I think it is actually fundamentally different from when we IPO'd, because we've worked on so many other different areas that are very important from a commercial perspective.
Excellent, thank you. If you can, another question here, if you can successfully identify the profile of patients who will be susceptible to cytokine storms,
yeah,
do you envisage an OBD blood test that could be used on the general population to prioritize who will receive a vaccine?
That's a good question. So that is a thought, and it's a very sensible thought. I think we'd rather comment on that when we let's wait until let's wait for a couple of months, but it is a thought. It's a very sensible thought.
Yeah, okay, makes sense. Are there any similar companies or examples of similar companies to OBD being bought by bigger players?
Yes, so I mean the best example of a platform company being bought would be Foundation Medicine, which again is on our slides, which was bought by Roche. It was a multi-billion transaction, and the reason that Roche bought them was that they believed that they would get stratification and also target identification for their drug discovery. So yes, the peer group transactions are very encouraging.
Yeah, okay. I think we've just got one final question now, so I'm going to give that to you here. And it says, what key milestones would you sort of flag out for investors to be aware of? And which of these will prove a successful direction of travel regarding the commercial evolution of the business?
So I think there are 3 milestones on the horizon. One is some reporting on our Glasgow COVID work. The second is, as was talked about before, was to do with the our contract that we signed in December and the work that we're currently undertaking. And the third is, which will be very visible on the Mitsubishi site, the delivery of blood to us for day 1 take and the Mitsubishi program.
Thank you. We've just had one last question, which I think we've probably got time for, and it's back to COVID-19, and it sort of said it. Look, COVID-19 is a very high-profile area, and Oxford Biodynamics hasn't had a historical record in infectious diseases. So why would you take this risk to enter this area? And I guess if it fails in COVID-19, will that not hurt your reputation of the company and the technology?
That's very good. So we think what we're seeing with these CCS is fundamental. So we don't think we'll fail. Otherwise, we wouldn't have embarked on this. And the reason that we have some faith in that is that we have worked in similar stratifications, as I showed in the presentation, in other areas. So we're pretty confident that we will be able to stratify here.
Christian, that's great. Many thanks. That's the end of the questions that we've had coming in. And so this brings us to an end to the webinar with Oxford Biodynamics today. Thank you very much for attending.